Good morning, everyone. Let's start the last day of the talks, and I'm going to introduce now the first speaker of today, which is Stephanie Castillo, with the talk entitled "Investigating the Evolutionary History of the Corsairs." Please welcome.
Thank you, Renan. Thank you everyone for joining me this morning. I'm excited to share with you what I have been working on as part of my dissertation at the University of California, Riverside. I have to look closer? Okay. So my research focuses on the Reduviid subfamily Paratini, which is the 6th largest subfamily of assassin bugs, with 32 known genera and over 340 species described worldwide. They are generalist predators, hunting mainly at night on the soil surface, where you may often find them covered by fallen plant parts and beneath stones. However, you don't want to mess with them when you do encounter a paratine, because species of paratini reportedly inflict the most painful defensive bites among reduviids. And as you can see from the pictures that I used on this slide, many species of paratini have bright colors such as reds, white, yellow, and even metallic blues and greens. However, one of the diagnostic characters for this subfamily is asymmetric male genitalia, which is very easy to spot. So this is what nearly all 7,000 described assassin bugs typically look like. So they're symmetric in terms of their abdomen and their pygophore. And so while general asymmetry is rare in Reduviidae, in Paratini we see here that the median process of the pygophore and the perimeres slant to the morphologically right side of the body. Additionally, certain males also have an asymmetric median abdominal carina on the abdomen on the ventral surface. And other males will also have these unique spine-like or hook-like structures on the 7th mediosternite, and a couple species of Fusius also have these dentate or teeth-like structures on the 5th mediosternite, and both of these structures are only found on the left side of the abdomen. And so those structures are very interesting, but we don't really understand what they do in terms of mating. But we do know that Reduviids copulate in a dorsolateral mating position, where the male starts off by mate guarding on top of the female, and then he slides down to the side of the female, where he then extends his genitalia and achieves connection. However, what we see in paratines is a bit different. So paratines are the only reduviids that engage in an end-to-end mating position, similar to what we see in most pentatomomorpha, which is interesting considering that males have asymmetric genitalia but engage in this seemingly symmetrical genital contact with the females. And so this is what the females look like. We're going to take a look at the ovipositor of Pyrotis stridulus and Cirthenia stria, which are interesting among reduviids because at the base of the first valvulae, as you can see here, there is a notch and a groove, and also these dentate ridges that appear to interlock, which could suggest that there may be some female choice occurring, which could make it more difficult for males to access females. So this is something that we don't see in other reduviids that we know of, and even and it hasn't even been well documented in paratini either. So it's a better way to understand this would be to put it into an evolutionary context, and so I decided to pursue some phylogenetic comparative methods, which I will share with you in a bit. Unfortunately, relationships across paratini are unresolved, so large molecular data sets such as this one show that paratini is monophyletic and one of the early diverging lineages of higher Reduviidae. As you can also see, though, these higher level studies are limited in their taxon sampling of paratini species and genera. And even the few studies that have explored these lower level relationships of paratini only focus within a few of the speciose genera, and they also have limited taxon and molecular marker sampling. It's therefore no surprise that the monophyly for most genera remains untested and that relationships among paratine genera are unknown. It also doesn't help that the taxonomic literature is surprisingly light on making assessments that we can turn into testable hypotheses, which just further highlights the fact that molecular phylogenetic analysis of paratiny is long overdue. And so my research objectives are to infer the first robust hypothesis, phylogenetic hypothesis across paratiny using ribosomal, mitochondrial, and anchored hybrid enrichment loci. And then I will use this phylogeny to reconstruct the ancestral character state of color patterns as well as male and female morphological diversity. And so my phylogenetic hypothesis is based on 15 paratine genera, which is the largest sampling to date. I then used a combined matrix with Sanger sequence data and 294 AG loci, which was then used to infer a Raxamel tree. I then used this phylogenetic hypothesis to reconstruct ancestral character states using parsimony and mesquite. And so having both the AG and the Sanger sequence data allowed me to combine a data set that was taxon-rich and character-rich to create a phylogeny that is well-sampled with high backbone support. I found that missing data did not affect the tree topology, and that these 9 taxa here with the red arrows, oh yeah, there we go, that these 9 taxa with AG data did not cluster together in the combined data set, and the addition of Sanger data did not change the relationships that were inferred from the data with the matrix with just the AG data alone. I also recovered 2 clades, so the bottom portion with an old world clade. Let's see if this mouse works. Will it show on the screen? Yes. And this other clade here with New World taxa, as well as paraphyletic Certhinia. And so I also covered that many monophyletic genera, which were highly supported, but some of the more speciose genera are not monophyletic, namely polyphyletic Ectomachris here in this dark blue, as well as paraphyletic Rhazahus in teal, and then paraphyletic Certhinia in pink. And so I hypothesized that this could be due to the use of misleading homoplastic characters to diagnose these larger genera, like the length of the fossila spongiosa or even the length of the head, for example. And so having this phylogenetic hypothesis, I was then able to reconstruct color patterns. And so the most recent common ancestor of paratiny was recovered as cryptically colored, or drab colored, as we see here in this Certhinia species. And so what I mean is that there is no iridescence or spotting of the hemelytra. However, many species of paratiny are not drab, and that's why I want to highlight these 4 color patterns that we will talk about some more. And so here we are going to look at the evolution of putative aposematic coloration, which evolved at least 11 to 13 times. I coded red, orange, white, and yellow patterns as aposematic, even though aposematism in paratiny has not been verified with predator avoidance tests. However, because these colors have been shown to be effective in other Heteroptera to deter birds and other visual predators, such as jumping spiders, I refer to these colors in paratiny as putatively aposematic. Meanwhile, iridescence with a blue or green hue on the thorax and or abdomen evolved at least 2 to 3 times independently within the Old World, but potentially more times since the iridescence is also found in New World taxa, which were unattainable for my study, unfortunately. I also reconstructed the evolution of a stripe and spot on the membrane of the hemelytra, which is a combination that is not found in other reduviids that we know of. Yet this pattern has evolved several times across several genera of paratiny, so at least 4 times in the New World and 4 to 5 times in the Old World. However, this sometimes the spot can occur without the stripe, or the spot can occur on several areas of the wing. So I reconstructed the presence of a membranal spot and found that it has evolved 1 to 5 times in New World paratiny and 5 to 7 times in the Old World. And these spots were not observed in Sardinia nor any Reduviid outgroups included in my analysis. And I find that the membranal spot is particularly interesting because it occurs in different genera from different geographic regions. So my hypothesis is that this membranal spot may function as eye spots to startle predators, also known as a dymatic display. There could also be mimicry rings with wasps, such as Calopompilus or Mutilids, that we are not aware of, potentially. And what I think is less likely is interest-specific signaling. And the reason I don't think that that this is the reason is because paratines are nocturnal and visual communication is not common in Reduviidi that I know of, at least. Therefore, I think that this pattern is more likely implicated in defensive encounters, but researchers would have to investigate this further to determine its effectiveness as a signal against potential predators. But unfortunately, we also don't know what is preying on paratiny. So we can only make guesses until further work is done. And so my next objective was to reconstruct male asymmetries, specifically the extragenital process on the 7th sternite, the asymmetric median abdominal carina, and the unilateral teeth on the 5th mediasternite. And all of these characters are absent in the large clade that includes the New World taxa as well as circumtropical Sardinia. However, this Old World clade down below, if the mouse is working, yes, it is. So this is where we see multiple independent origins of these characters. So in one instance, we see that the extragenital process occurs by itself, but in 7 species, abdominal asymmetry co-occurs with either the extragenital process or the lateral teeth. And more specifically, the lateral teeth and asymmetric abdomen evolve together in one species, but in all other instances, the asymmetric abdomen evolves before the extragenital process in 6 species within 3 clades. And when we look at this larger clade here, wait, if I can get the mouse to work right here, we see that only some of the descendants with an asymmetric abdomen end up gaining the extragenital process. So based on these patterns, we hypothesize that abdominal asymmetry makes it more likely that an extragenital process will evolve in some of the descendants. All right, and finally, for the females, I found that there are multiple independent origins of interlocking structures on the ovipositor of paratiny. So Reduviids typically have this simple valvulae, simple valvulae without any interlocking modifications, but within paratiny, these longitudinally grooved valvulae evolved once in Oriental Sardinia, and then the dentate valvulae evolved 1 to 2 times in New World taxa, and this notch and groove evolved once within the Old World paratiny. And this is the same clade where we saw multiple independent origins of the abdominal asymmetry and extragenital processes, so that's pretty interesting. And there have been multiple transitions back to a simple ovipositor, but I must note that a limitation to my study is that female paratines are rare in collections compared to males, which means that my documentation of females is limited because, and that's indicated by the missing data of these gray bars. But I still hope that this analysis is enough to highlight that there may be potentially interesting mating behaviors selecting for these unique structures that we are not aware of yet. All right, so I'm going to conclude today's talk by summarizing the main highlights of my project here. And so I have inferred the most densely sampled phylogeny of paratiny to date, and it highlights the gaps that need to be addressed, specifically in the large non-monophyletic genera. So there could be some classification updates that need to be done. I have also found the convergent evolution of color patterns, for example, the putative eye spot, that could be involved in defensive signaling. And lastly, I found the independent evolution of various male and female genitalic traits that could imply that the interactions between males and females of paratiny are more complicated than what we currently know. And so with that, I'd like to thank you all for your time and everyone that made this study possible. I'll be happy to take any questions or hear whatever you have to say about this, so thank you.
Are there any questions?
Matt.
Great talk, Stephanie. I very much enjoyed that. I have a million questions, but I won't do them now. Yeah, I've got one question about the aposematism idea. It's really cool. If you were to suggest that it was aposematism, what kind of receivers would you expect? What kind of predators do you think would be responding to that? And also, the coloration is in the adults, but do you see that in the nymphs as well, by any chance? There's a button at the bottom.
We do see aposematic coloration also in the juveniles or the nymphs. They are, typically red or iridescent, but obviously there are no wings where we can see any patterning, but we do see that. And then the first question was about
a potential predator, because, yes.
So I think it's probably going to be visual predators like birds or lizards, something that's along the ground floor, kind of pegging around through leafy litter, since they're often on the ground.
Okay.
That's my best guess.
All right.
Are there any other questions?
I find it interesting that, in some groups there is no male genitalia, and here you've found that it evolved several times in this group. Any speculation as to why this group is more susceptible to development of asymmetric genitalia?
Yeah, that's a great question. So I think that it could be that change in mating position. I think that's enough to kind of there be selective pressures on, like, kind of accommodate for this mismatch or this compensation for between the left and the right parameres to try to compensate for this new mating position and this switch that is only in paratines and not the other renovioids. So.
Thank you.
Anyone else?
Do you think, concerning the aposematic coloration, that they are advertising for some of the distastefulness, or rather for the painful bite?
Yes, actually, that's a great question. I do think so as well, yeah. I mean, they supposedly have really painful bites. I keep hearing that over and over again. So I think in a way they are also advertising their defensive bites, yeah.
All right, thank you.
Good morning. I will not talk about any taxonomy questions, but I will talk about one specific species that entered, is an invasive one, and entered in Spain, like in another part of the world, and how it is dispersing inside our territory, and also the consequences that this has. I will not talk anything about now Alliomorpha, because yesterday we addressed this topic in several other communications. So this is the current distribution that the last map that the European and Mediterranean Plant Protection Organization released. And as you can see, the species is most distributed in the Northern Hemisphere. It was only found in one country officially from the Southern Hemisphere, which is Chile. And there are several studies regarding the invasion since the point of view of genetics, and it was demonstrated that several invasions occurs in between the countries, not only from the endemic area, but also in between the countries that already has the species. And in fact, in the north of Italy, they demonstrating that they still has invasions. So it was not only one invasion, it can be several ones from several origins. The colleagues of New Zealand, which you know is one of the countries with more strict quarantine measures and protection of their borders, made a study in which they demonstrated that in which kind of goods it is possible to find Alliomorpha allis. And they showed that the most frequent good carrying Alliomorpha allis are new vehicles. The second one was used vehicles, and the third one was boots, trunks. So we can clearly say that Alliomorpha allis travels by car. So, in fact, in the north of Italy, they have demonstrated that at the beginnings of the invasion, the distribution of the species is following the main roads of the country. So, in our case, I will show that it's quite similar. So, if you visit the site of the CABI, they have this very good description of the pathway vectors of Alliomorpha allis. It can travel by aircraft, by boats, in a container, in a suitcase, really in any vehicle. So each of us can be vectors of Alliomorpha allis, because the behavior of the species, which used to overwinter in human-made buildings and likes fabrics, it is quite possible that you have, if you have in your country Alliomorpha allis, you can have in your wardrobe. So you have a cloth, you fold the clothes inside your suitcase, you take a plane, and when you arrive to another place, you took with you Alliomorpha allis. So all of us are vectors of Alliomorpha allis. So I will show you this other characteristic of Alliomorpha, which is the aggregation. This is a picture of trees in our province, in Girona, the northern here, 100 kilometers to the north of Barcelona. And this is in Italy. I will show it because it's well known because, let me see. All these dark points are Alliomorpha allis, trying to enter in a house. This is in Friuli, in Italy. And this noise is a brush brushing alliomorphs, hundreds, thousands of alliomorphs in the soil. So you can imagine how the nuisance that this species produce in the people is really very complicated. So the species lives in the three environments, in the trees of cities, in natural environments, and also in agricultural one. So it's highly polyphagous. It was cited in more than 100 species, but we certainly know that 300 or 3,000 can be, because we have a list of plants that has not been cited yet, that we found it living and reproducing. So the first record in Spain was in the year 2016. It's a funny history because it was an Erasmus student coming from Italy that was in the Girona University. He was aficionado to entomology, and he collected close to the place he was hosting some insects and come back to his university in Italy, and the professor that review the species identified clearly Alliomorpha alis. They were very kind and they informed us that they found the species here, so we informed immediately to the authorities because we know what was happening in Italy. It was very dramatic, the situation in crops in Italy. So at that moment, we immediately start to collaborate with the University of Barcelona, where the most important specialist in Pentsatomidae, and also in Hemiptera in general. So we gather the data that we have at that time of the distribution in 2016 and 2017, and in 2018 we published the first study on the distribution of Alliomorpha alis. This is the European Union, this is Spain, and this is Catalonia, and it was found for the first time here in the northern part. So, as I said, we published this study in 2018 in which we already find Alliomorpha in Barcelona, in several places, in several other municipalities surrounding the city of Barcelona, but it was very curious that these municipalities were very close to the port and the airport. So we established at that moment the objective of knowing the distribution in the space and over the time of the species, and also to know the response of the species to commercial aggregation pheromones to use it in monitoring, because we want to know how we can know where was the species. So we started to do several activities. The first one was to open a web page in which, for collecting also observation of the people, we started the project of citizen science named IHS Wanted in the Natosfera platform, which is a naturalist platform that you can open several projects inside, and we did it. And the requirements for the people was to include a clear picture of the insect, mainly in dorsal view, include the municipality and the geographic coordinates, and something happened here, and also they have to indicate where they found it, if they found inside the house, in a tree, in the nature, as much details as they can. And also we included a link to the official site of the species that is embedded inside the official webpage of the Department of Agriculture. So this is the link to the webpage. We also review other naturalist platforms, because there are many aficionados to entomology that take pictures and hang the pictures there. We also collected all the official reports, because the government was funded all these studies and gave us all the official data. And we also installed installed several monitoring traps baited with the dual lure that was already produced by the USDA in the United States, because they are dealing with this pest for more than 12 or 15 years now. And the dual lure has the pheromone produced by the male of Aleomorpha alis, plus another pheromone produced for other species, which is Plautistali, which is synergist, and it's proven that it's the best for the species, attracting the species right now. And we use two types of traps. This is the pyramid traps that is used mainly in crops, and this is the, I do not know. No, I do not remember now the name, but it's the coed trap, and that is mainly used in cities, at least here. But both traps are has to be imported from the US, because we haven't here a trap for this kind of insects. So the results here, first, regarding the citizen science, we, in this period, we collected 1,130 valid records, so which is quite a good number of records, and the mainly 69% coming from our project, HH Wanted, and also we found the 70% of data from eNaturalist platform, and also from Biodiversidad Virtual, which translating is virtual biodiversity. It's another naturalist platform, and in less quantity we receive by email. Many, several citizens send us also emails, and also social networks that we review, mainly Twitter. Twitter has information about the species. One other thing, the citizens not only send us pictures of the species for identification properly, but also they send us pictures of the number of the insects that are trying to enter in the houses. This is in Barcelona. You can see this curtain with a lot of insects trying to enter in the house, and this is in Girona in a window. Well, with this data, we built several maps that you can see. This is 2016, 17, 18, 19, 2020, and 2021. There are two 2021 because these are municipalities. We paint municipalities, and you can see mainly is close to the sea border, but it's going inside in the year 2029. We found in the province most farther of the sea here is Lleida province, in which is the main area of fruit cropping of Catalonia. So it was scary when we found there, because we were waiting that it will not arrive here because it's driest than these other provinces. But if we see, the counties, we can see that all the counties of Girona are already colonized, and mainly of the counties of Barcelona, and also the counties of Tarragona province, where hazelnuts are cropped, and hazelnut is also a very good host for Aleomorpha alis. And in this map, you can also see at the beginning, these dark points are the main roads, and we can see here because the municipalities were officially communicating they have it, and mainly of the municipalities were close to the main road, which is the high road coming from France, the AP7. So the pattern that it was identified in Italy is similar here. So if we analyze the number of municipalities that were informing they have the species year after year, we found an exponential pattern. So each year, the municipality having Aleomorpha alis multiplied by 10. But in 2021, this pattern broken. And we didn't, because we expected last year that we have a huge population, a huge problems, but it didn't happen. So this was because the weather, and this was the first time we find some cues that can control a bit the level of the population. In 2020, we have very low population, both in Barcelona and Girona area. But in Girona province, in some areas, we have some microclimate and there were problems with the fruits. So if we see here the monthly citizen report, we can see that October is the significantly higher number of reports. And this is because at that time, Aleomorpha alis is entering houses. But this also gives us information about how functions citizen science. We have not all the months equally sampled. So, and this is the situation in Spain. We start to collect also information from other communities in Spain in our webpage that it was open at the beginning only for Catalonia, but you can see here that at the end of 2021, we have in the Basque Country, in Pamplona, which is Navarra community, in Madrid, and in several municipalities, also here in Extremadura, in Andalusia, in Sevilla, and in the Valencian community, in both provinces, in Valencia and Castellón. So, but it's curious that here in Aragon we haven't, but this year I have to say that already found here. So finally, this is the pattern of the monitoring in traps. You can see that the pattern is different in immatures and in adults, that the main immature were captured in June and the main adults was captured in September. So this is giving us a lot of information about how is functioning this attractant. It's an aggregation pheromone. It's not a sexual pheromone, so it doesn't serve, or it's not useful for monitoring the species all year round, because when Aleomorpha alis is induced to overwinter it, any stimulus is attracting the species. So it's very complicated to capture at that time. So I will, because they said, well, okay, thank you. So because I will stop, but there are only reflections. The main one here is that much more knowledge on the biology of the species and also on the behavior is needed to find the weak point for controlling it, and we need to control it in all the environment. So we need to develop more than one control method, because in citizens here, in cities here in Europe, many chemicals are forbidden, and in natural environment also. So we need to find several solutions to combine them for controlling in all the environments, because it's not possible to only control in the agricultural environment. We have always invasions from the other environments. Finally, I want to let you the pictures for knowing the measure of the tragedy. It is really big. So this is pears, tomatoes, peppers, several varieties of apples, and also olives. I can also put here pictures of grapes, picture of kiwi, because kiwi likes a lot. Also from soya. Soya likes a lot to Aleomorpha. So, but well, these are damage from our orchards, currently damaged. And finally, I want to tell you that we are organizing a big meeting of two groups, working group of the IOBC in Girona in September, and for sure it will be a complete session of Aleomorpha alis, We have received almost 20 abstracts already for this session, so I hope we can discuss with all the colleagues how we can advance in the control of that species. And thank you very much.
Thank you. And if you want, someone has some questions, please ask her during the coffee break. We don't have time, unfortunately. Thank you. We're entitled Untangling the Web, Systematic Stem Biogeography of Translated Assassin Web.
Yeah, try not to move the computer just in case this moves. And good.
Oh, awesome. Thank you. How does this, is it? Ah, great. Okay. Oh, I need to, can I just like, can I hold this? Okay, well I can just hold this, I don't mind. Yeah, I'll just do this. Sometimes people tell me I'm too quiet, so I'm really excited to talk to you today about my research on Emicini, the thread-legged assassin bugs. So Emicini are one of the largest subfamilies of Reduviidae with around 95 genera and over 950 species. They exhibit a wide range of feeding behavior, some of which we were lucky enough to hear from Anne Wignall about a month ago. And this includes obligate spider predators like the Emicine nymph on the left, and also kleptoparasites where they will feed on prey caught in the spider web, and then also free-living species like the Emicine on the far right. And these free-living species don't appear to have any association with spider webs at all. Emicini are also really unique among Reduviids in having a cosmopolitan distribution where there are 4 genera that are found around the world, and then 13 species are found on more than one continent. There are also numerous cave-dwelling species, and there are numerous island endemic species. And it's interesting because most Reduviids are not found on islands, and so actually if you're collecting on an island, you're more likely to find an Emicine Reduviid than any other Reduviids, which is in contrast to normally when you're collecting, you don't really find many Emicines. So it kind of lends itself to this question of how are they dispersing so well, especially when you think about their wing types. So you can see there's an Apterus species of Emicine on the top in the middle. Also, it's maybe a little hard to tell, but this Emicine, the wings end like maybe 2/3 down the abdomen. So there are several Brachypterus species that have very small wings, and then even the species that are fully winged are just not good flyers. Emicini, they're not dispersing based on their flying ability, that is for sure. But they are found on logs and hanging leaf matter. And that has kind of lent itself to this hypothesis that they're floating on rafts on ocean currents to different continents. Throughout my talk, I'll be referring to Emicini sensulato, and I want to briefly introduce these 3 subfamilies that are forming this group. So Emicini have this extremely elongate forecoxa that's at least 3 times as long as it is wide, where Cyacini and Visaenocrine have a much more typical coxal length. Emicini, you can tell by their common name, the thread-legged assassin bugs, they have these really long and skinny mid and hind legs, where Cyacini and Visaenocrine have much more typical leg structure. Then looking at the labial segments, Emicini have more typical labial segments for Reduviids, where Cyacini has this expanded second visible labial segment and sometimes a third as well, and then Visaenocrine has this extremely elongate first visible labial segment. Looking at the phylogenetic hypotheses within this group, in 1966, Wygodzinski looked at relationships and found that both Cyacini and Emicini are monophyletic, with Visaenocrine coming in sister to Cyacini, and Visaenocrine and Cyacini together sister to Emicini, forming Emicini sensulato. In 2012, Wang and Weirauch looked at relationships across all of Reduviids and include some taxa from these 3 subfamilies, and their results did corroborate Emicini sensulato. However, if you can see here, Visaenocrine is coming out in the middle of Emicini, kind of giving us this idea that we really need to pursue further to understand better the relationships between these 3 subfamilies, and also tribal level relationships within the diverse Emicini were not tested. So my objectives for this study were first to reconstruct a phylogenetic hypothesis of Emesani sensu-lato, resolving the relationships between subfamilies and tribes of Emesines, and then, based on that phylogenetic hypothesis, to revise the tribes within Emesani sensu-lato and generate diagnostic characters using an ancestral character state reconstruction of a morphological matrix based on my molecular vouchers. And then finally, I wanted to investigate more these 4 cosmopolitan genera and their biogeographic history, and so I generated a divergence data phylogenetic hypothesis to estimate their biogeographic history. So for my first objective, I used a combined data set with anchored hybrid enrichment and Sanger sequencing. And we'd really tried to sample carefully for this AHE data set, including representatives from each subfamily and all 5 Emesine tribes. And then the Sanger sequencing data set was much more deeply sampled with 155 in-group taxa. And I aligned everything using MAFT and ran my trees in IQ-tree. And here you can see the results. So I want to just point out that those purple stars, that's showing where those AHE loci were. So you can see that they're not all clustering together, similar to in Stephanie's talk, and that they're spread relatively well throughout the tree. Some of it's weirdly formatted. But you can see here that Cyacine is paraphyletic. This down here is a genus Oncerotrachelis that's coming out sister to the rest of Emesine sensu-lato. And then also we have up here Metapterini, this tribe, and Emesine, the other tribe, are both paraphyletic, forming this large clade here, with Ploia arleni being monophyletic, but nested within those 2 tribes. So then going on to my revised classification, the main kind of goals that I had for this revised classification was that this would be both monophyletic and easily diagnosable. So I chose 40 morphological characters based primarily off of Y. Godzinski's 1966 monograph, and I generated these characters using my molecular vouchers. And then I ran ancestral character state reconstruction in R using maximum likelihood. So another, debate that I had in my mind when doing this is how am I going to split these things up, right? Me and Christiane went back and forth a lot trying to decide how to split these 3 subfamilies, how to split them back up into groups that made sense. So we kind of discussed having 4 different subfamilies, 3 subfamilies, 2 subfamilies, one that's very big and then one very small. Or finally, to just have 1 subfamily with 6 tribes, and that's what we ended up deciding to do for our future manuscript. So now I'm just going to briefly touch on some of the morphological characters that will be used to describe these new tribes. So we have this combined Emesine, Metapterini, and Ploia arleni group that was shown earlier, and you can see that they have this really, really interesting structure on their 4 tarsal claws. They have this comb-like structure here, and then they also have this notch that's found on those 4 tarsal claws. And we're not sure, but given that many species in this clade are associated with spider webs, including Stenolimus, we think that they might be using this to manipulate the spider web in different ways. Also, the ventral surface of their foretibia has tuberculate setae, and on their forewing, they're missing this MCU AN1 cross vein that is frequently found in other groups. Then looking at Cyacine, excluding Oncerotrachelis, we have the second visible labial segment is swollen. Also on their forewing, the colors are maybe a little hard to see in this presentation, but here the M and CU veins are separate along the proximal portion of the wing, which is pretty unique in this group. And then I love this SEM, and it's a little hard to see here, but the distal tarsomere's ventral surface, they have these really cool looking flattened and widened tendon hairs that are not found in any other groups in Emesani sensu-lato. And then Oncerotrachelis, they have on the postocular portion of their head, it's twice as tall as it is wide, it's swollen, almost rounded, and raised above the anteocular portion. And then also looking at the ventral surface of the labium, on their first and second segments, there's some pretty thick macroceti present. And then looking at the forewing, you can see right here, it's pretty small, but the R and M vein are fused along the proximal portion of the MCU cell. And then seeing that purple highlighted region there, this MCU AN1 cell, that is only found in Oncerotrachelis. So now going on to divergence dating, I ran my divergence dating analysis in BEAST-2 using the birth-death tree model and the relaxed log normal clock model. And I also used a fixed ultrametric tree just because my data set was too large to complete without that. And then I ran biogeobears in R, and I chose the DEC plus J model as it was the most highly supported model out of all the ones I ran. And then I included 6 fossils covering 2 tribes. And just to briefly touch on these fossils, Christiane mentioned yesterday that Emicines are remarkable among rejuviids for having 13 fossils, which is way more than any other subfamily. However, these fossils are mostly all in one tribe, and they're all about the same age too. So I was only able to include 6 because of that. But still, that is a lot of fossils for rejuviidae, and I was really excited to have so many. So here you can see my results. These little skulls are showing where the fossils are placed. And you can see that they're pretty heavily biased towards Polariline, but still I was able to get some others that placed elsewhere. So just briefly going over my divergence dating results, kind of the broad picture. During the Upper Cretaceous, we had Emicine sensu-lato diverging from the rest of Rejuviidae. We also had this large clade Emicine metapterine pluriline right here, diverging during the Upper Cretaceous. And then Cyacine, excluding Oncerotracheus, was also during the Upper Cretaceous. And then during the Eocene, we have both Leistarchine and Colartodine diverging. And during the Oligocene, we have Oncerotracheus and Visianocarine diverging. And then here we have highlighted these cosmopolitan genera found throughout the tree. Oh, and I wanted to know, these here are the range, right? So I'm giving the average age, but there was definitely a wide range for plenty of the nodes. So during the Oligocene, we have Gardena, Amphicarus, and Ploeria diverging. And then during the Miocene, we have Stenolimus diverging. And I'll go into those in a little more detail during the biogeography portion. So kind of touching again on this high dispersal ability, there are 4 genera of Emicine again that are cosmopolitan. And this is very rarely found in other Rejuviid subfamilies. And so one question that was tossed around for a long time was maybe these genera aren't actually monophyletic, and maybe that is how they're found all over the world, is they're not, true monophyletic groups. And because again, they're not very good flyers. So I really want to look more closely and see if these groups were actually monophyletic. So for these biogeobarrier results, here these squares are showing the maximum likelihood discrete states. I was not able to show all of the possible options for ancestral areas just because that would have been very difficult to portray in a presentation. However, I found that the neotropics are ancestral for Emicine sensu-lato, and the Afrotropics are ancestral for Emicine, so for everything excluding Oncerotracheus. Sorry, Afrotropics and neotropics, a combined range. So here, looking at Gardena, and I'm just kind of going to step through each of these nodes. You can see here that the ancestral area for Gardena was a shared range between Afrotropics and neotropics. Again, just showing their remarkable dispersal ability despite their lack of flying ability. Then we have some speciation within the Afrotropics and neotropics. And then further dispersal from the neotropics into the Nearctic region and from the Afrotropics into Indo-Malaysia. Then looking at Stenolimus, we have a shared ancestral range between Australia and the neotropics. And I want to really emphasize here that these results are heavily biased towards our sampling. We have mostly taxa from Australia included, with 1 from the neotropics. And this genera is found all over the world, but we just did not have samples from all over the world. So really, to get a better look at this, we'd have to have an increased sampling. Now looking at Amphicarus, I found that Amphicarus had an ancestral range but shared between Afrotropics and Indo-Malaysia, with then further speciation within the Afrotropics, and then dispersal to the neotropics in this shared range. And then we have also further dispersal into the Palearctic region and speciation within the neotropics. And then further dispersal into the Nearctic region and speciation within Indo-Malaysian region. So now moving on to Ploeria, I didn't show in detail the other genera because they were all monophyletic, but this one, maybe you can barely make out these names, but I found that Ploeria was not monophyletic, and this I mean, I was not able to sample the genus fully or anything, but I just wanted to add that caveat that this genus was not monophyletic. It had an ancestral range of the neotropics, with then some further speciation within the neotropics, and then also dispersal into the Indo-Malaysian region. And then we see dispersal from the neotropics into the Nearctic region, and then also dispersal between the Afrotropics and Indo-Malaysian region. And then we see further speciation within the Nearctic region, and then also dispersal to Australia, and then further speciation within Australia. So in summary, one of the kind of main takeaways from my research was that really Emesani sensu-lato clearly needs a reclassification. I found that Sayasini was paraphyletic, with both Bisayanocrini and Sayasini rendering Emesani paraphyletic, and 2 tribes within Emesani were not monophyletic. I also found that the dispersal history of this group is really complicated, that clearly they are very incredible, they have a very incredible dispersal ability, and 3 of those cosmopolitan genera diverged during the Oligocene from different places, and 1 genus evolved during the Miocene. However, I really need increased sampling for Senalimus if we want to get a better view of their biogeographic history. And then I also found that Ploieria is likely paraphyletic. They are very, very hard to ID, so there could be some identification issues there, but I've also gone over them several times, so increased sampling is need to really determine that. And with that, I would like to thank all of the lab members, past and present, and my advisor, and all of our funding sources, and I'm happy to take any questions.
Thank you, Samantha. Questions?
Thank you for this incredible talk. I want to ask, so what about Collartidini, which by many morphological classifications were considered to be the sister to the other Emesine tribes? I was just wondering what the topology was, because I couldn't clearly see it from the presentation. Thank you.
Yeah, it was pretty hard to see in that big tree. Collartidini is sister to Bisayanocrini, which I'm assuming you've looked at both of those groups, and they do share a lot of morphological characters, so I felt pretty comfortable with that result.
Anyone else? Okay, thank you so much.
Good morning. I will talk today about a project of West European fauna of Myridae in the series of book Faune de France. And it is this series has nearly 1 century, is nearly 1 century old now. So I did a talk a long time ago in Wageningen in 2006 about the series Faune de France, but I will give you a few informations because probably that a lot of people here were not there a long time ago. And the Faune de France is a series of books created in 1921. The first volume was about Echinoderma. And the last book was published a few weeks ago. It is about Hymenoptera. And so the Faune de France is more than 100 years old. The editor is the French Federation of Natural History Societies, and it is a not-profit-making association, and sales of the books is the main resources. The main purpose of that books is to give tools to naturalists and scientists who want to identify and study animals in France. At the beginning it was restricted to France, and now we need France, of course, but we the geographical area covered are usually wider now than France, and you can see on the map extracted from the book about Choreoidea that we are coming from nearly Ural Mountains, and usually we included also North Africa. The Heteroptera is a very well treated taxonomic group in the Fond de France with 15 books, and thanks in particularly to Jean Péricard, who was the director of the Fond de France from 1983 to 2003. And during that period, he wrote 10 books about shoe bugs, an amazing amount of work. So this is a fictive phylogeny, just to show you what we did in Fond de France about European Heteroptera. The first book was written in 1957. It was about aquatic insects, aquatic bugs, Gyromorpha and Nepomorpha. It was written by Poisson, Raymond Poisson. The second book was about Murid by Wagner and Weber. Then we have a series of books written by Jean Péricard. The first one about Tengidae, then Beritidae, Nabidae, Saldidae and Leptopodidae. Choreoidea was written by Pierre Moulet. Then 3 volume, very important volume about Ligaeoidea from Europe by Jean Péricard. A first book about Pentatomoidea by Valeriu Derginski and Jean Péricard. And then a book that covered Dipsochoromorpha by Ice and Péricard. The same book covered also Aradids and Piesmatids. Reduvids were treated by Pouchkov and Moulet. And another part of Pentatomoidea by Péricard, and the last one by Jordi Ribes and Pagola-Carte, that is present here. So, at that time, we have nearly covered all Heteroptera in Europe, except Scuteleridae, Acanthosomatidae, and Piesmatidae, that are in progress by our colleague Attilio Carapezza. And Anthocoridae were treated by Jean Péricard. It was his first book in 1972, but it was not in the series Fond de France, it was another editor's. But the book is available, and so nearly all the phylogeny was covered by books in Europe, except Tomastocoridae, but it's not really important as we have only one invasive species, so we don't need a book to identify it. And Cynid is the last group for which we don't have at the moment an author. So, to summarize, we have 15 books about Heteroptera that represent 6,200 pages. 7 of these books are available for sale, and I have a few copies I will sell at the coffee break if you are interested. And 8 books are out of sales and will be not published again, so we provide freely the PDF file on our website that is indicated below. You can get the PDF files, and you can get also the PDF files of the book about Anthocoridae by Jean Péricard. So, we have a new project of book, and it is about West European fauna of Meridae. Why we want to do a new book? We have already one, but this book is ancient. It was written in 1964, and since at that time it was a very important and very interesting book, but since 60 years, there is a lot of taxonomic changes, a lot of synonymies, and a lot of new species described, and so we need a new book. This book is badly illustrated. We have a lot of drawings, but we don't have a lot of habitus, and when you use the keys that are not easy, it's always difficult to know if you are in the good genus or not, if you don't know Mirids. And Fond de France are books for good naturalists, but not specialists. So they must be used by coleopterists, of course, and it is not the case. Someone that is not specialist of Mirid, it's very difficult for him to use that book. So we need a new book. It is very challenging because we have only one book to cover Fond de France at the moment, but Mirids represent more than 1/3 of the true bugs in Europe. And I give you the statistics for France. We have 511 species, more or less, of Mirid in Europe, in France, that represent 36% of the true bugs. So this is a very large group. This is a complicated group, and probably the most diverse and complicated family of Heteroptera in the world, and of course in Europe. So I would like to thank very much the authors who accept the challenge. Some of them are here in the room. The first one is Armand Matocq, probably one of the best specialists of Mirid in Europe, that is present here. If you want to speak with him, is here, from France. The second author is Frédéric Chérot from Belgium, a very good specialist of Mirinés of the world. Then Pagola-Carte Santiago from Spain, that is here, a good specialist of Orthotilinés, and myself to manage and organize the book. We need a lot of pictures. I think it's important to help to identification, and we are very lucky because 2 German people who are very good photographers accept to give us all their pictures. This is Gerhard Strauss, who published a CD-ROM that contains nearly all European species in habitus and genitalia. This is a very important work. And I would like to thank also Eckhard Wachmann, who did very amazing pictures of living specimens. So, we have some publishing constraints. We don't want to exceed too much 2,000 pages that represent 4 volumes of 500 pages each. And so if we limit each monography for one species to 2 pages, illustration included, we cannot cover more than 1,000 species. So to respect that constraints, we adapt the geographical area that can be covered. And you can see the smiley. This is the country that will be included in the book. We will include Spain and Italy, but not Greece, for example, because there is too much Mirid in Greece to be included. And at the end of the book, we will cover about 800 species. So the first volume will be written by Frédéric Chérot for the introduction of Miridae. And for the first groups, Isometopinae, Silapinae, Bryocorinea, Deraeocorinea, and the tribe Stenodelini of Myrinae, the authors will be Armand Matocq and I. The second book will be about Myrinae and other tribe of Myrinae by Frédéric Chérot. The third volume will be about Orthotilinés by Pagola-Carte. And the last one will be about Filinae. I think we will need 2 volumes because this subfamily is too important to be treated in only 1 volume. So, just to show you some part of the books that is in progress. This is a plate that was made by Frédéric Chérot to illustrate the introduction of Myridae, and this one is about the pubescence, that is an important character for taxonomy in Myriads. All the keys will be provided in French and English, and they will be illustrated by characters, like this plate for Adelphocoris. Each monograph will be very short, 2 pages max, with illustration and also with a small description of the species and very few and summarized information about biology and distribution. Distribution will be very synthetic. So we planned the following date. We hope to publish the first book in 2023, the second one 25, the third one 26, and the last one in 27. Up. At the moment, the introduction is finished because our colleague Frédéric Chérot works very quickly compared to other authors. And so introduction is very good and finished. We are working with Armand Matocq on Bryocorinae, and we have 2 huge problems because we have 2 huge and difficult genera in that subfamily, especially Diciphus. The genus Diciphus and Macrolophus. They are not easy to compile, but we have now a key to Diciphus, thanks to Armand Matocq, that will be really wonderful compared to what we have now. The second volume is nearly finished. We have 600 pages that was written by Frédéric Chérot. So perhaps it will be published before the first one. And we have also, the third volume is just starting, and the last volume will be start when the first one will be finished. So to conclude, I would like to say that Fonds de France is a non-profit association. The authors are not paid, and since 1983, sales have been sufficient to publish 1 book a year and to balance the budget. It's quite incredible, but it's like that, and it's a very exceptional way to work. And we hope to be in position to continue to publish books that allow reliable identification of European fauna, not only insects, we have also some mammals, and especially about true bugs. Thank you for your attention. And if you have questions, we will try to answer.
Are the islands included? Are the islands?
Yes, we will include islands from Spain and Canary also, and Corsica, Sicilia. So it's Madeira, yeah, yes. Yes, we included all the islands of the country included in the geographical area.
More questions? Okay, thank you.
Thank you.
Can you hear me like this? No. No, I have to take it in my hand? Oh, fuck. So, hello? Yeah.
Our next talk is called About the Others, Those Not in Your Bed, Ecology of Simicity, with Steffen and Roth.
Okay. Okay. Hello, everybody. Today I will talk about bed bugs, simicids, but not those which kind of come into your bed, disturbing your privacy and taking blood and causing nightmares. Maybe I will talk about a little bit more tender relationship. That is, the bed bugs are not using humans as hosts. But first, I would like to remind you that there are over 100 species of Simicidae with a wide range of different kind of morphological variation, but all of them are ectoparasites using different kind of vertebrates as hosts, mostly bats and birds. And all of them also are secondary wingless and have to use their host for migration or transport. Short outline of my talk. In the beginning, I will talk from the point of view of parasitology. What do we know from simids? And the second part, I will give some examples from mismatches of host distribution and parasite distribution using 2 examples. And finally, I will mention some population genetic studies in Simicidae. Okay, if you are a parasitologist, you are interested to know about virulence, the fitness, the parasitic load, infection rates, life history of hosts and parasites, and things like host selection and transmission rates. To study that in a proper way, you need to count. You have to know how many of your individuals, which are the host, are affected by the parasites, and then you even will produce a huge data set. You can do correlations, and you even may be able to manipulate the whole system. That makes, for point of view of a parasitologist, makes a organism as a idle study group. I was kind of baffled. Why didn't we learn so much more about the ecology of simicids, despite that bats are really intensive studied, and including ectoparasites of the bats? And the reason is, I think we'll explain here. If you enter a hotel room, then the bugs are already there. This is a common feature for the simicids. They are already in the place where their reproduction will start and where the host will come. Then they enter your bed, and then they go back on the wall. The same is in caves. So the bats coming, and then the simicids are there, and then they're entering the beds just for sucking blood, and then they go back. That is not a good thing to count if you want to know how much influence this parasite has on your host. If you catch the bat, you can say, okay, it's no ectoparasites on it, no simid on it, no affection, but it might just have a visitor which was sucking blood. So this makes it really complicated, because in a way you cannot count the simicids on your host, you have to count them in the surrounding. It is a little bit easier for birds, because here the simicids stay on the nests, and then you can relate the numbers of the simicids to the pair of breeding birds, to the nestlings, and so on. So, from point of view of a parasitologist, you should study birds if you are interested in simicids. And that is really the case. In particular, swallows are really good studied if it comes to the interaction with simicids. Okay, some results. In a way, is kind of, if you look for the European swallow bug, the Cimex hirundinus has been Oeziacus before. We know from a classical studies from the 1939 from a province in Germany that almost all nests from house martins were infected, but only few from the barn swallow, whatever the reason for that is. What do we know about these things today? Not much, because there's not a single similar study in any province in Europe done the last 50 years or so, and even it's a little bit more complicated, because if you look for the publications for the published records, you will find very few records for Cimex hirundinus in Europe. But if you talk to the people, they will confirm you. If you check house martin's nest, they are always there. I know this information from the Netherlands, from Switzerland, from Austria, and from Germany. Anyway, what you also can see, the number of individuals per nest can be high. Several hundreds, maybe up to 200,000 individuals per nest with Simicids. And this is easy to imagine, has a tremendous effect on the host. And from this long-time study from Charles Brown on the cliff swallows, we know that Simicids are parasites which really affect their host. So we have kind of a high nestling mortality, the shortening the breeding season, the reducing the survival of the host, they influence the metabolic growth, and we know a lot of transmission and dispersal from this system, and the transferring also viruses. So to sum it up, yes, Simicids, they are strong parasites which can affect their host, but the most things what we know is from birds. But there's also evidence that bats using a different roost because of the high Simicid load. Okay, in the next part I will show you two examples how the distribution of the host does not really match with that of the Simicid. First example comes from Europe. We have this Cimex pipistrelli, which is, according to molecular data, one species, but you know from the history they were considered as several species, so I call them pipistrelli complex. And what you can see on the right-hand side, that is the distribution of one of their main hosts, that is the great mouse-eared bat, and you can see it is spread almost all over Europe. But if you look for the map with this records from Cimex pipistrelli, this species is missing in the Balkan area and in Turkey. Nevertheless, that the host occurs there. And the study from Balvin also showed if you make like a transect from Central Europe to the south, you get less and less records. And I can confirm that because just 4 weeks ago, I found the first records for Cimex pipistrelli for Armenia and Georgia. So this species Cimex species getting rarer and rarer if you come to the south, even if the hosts are there. And one explanation might be this. The Cimex species occur in Central Europe, both in caves and in human dwellings. But if you go to the south, you only find them in human dwellings. That might be churches, old churches, ruins, also houses where people still live, as long as there are bats. The bats are not doing that. They are in caves all over in Europe, not restricted only to buildings. And interesting side, I think, is kind of the human bedbugs have never been found in a cave, at least not in Europe. So why can't that be? You have to keep in mind that the Simicids, they need a minimum temperature for development, which is kind of 14 degrees. They do not like too wet caves, and they also do not like too dry wet caves. And they have one problem. The Simids stay in the cave, then the host came, and then they can suck blood, and then the host usually, the bats in particular, overwinter in different places. So they have to stay there for months and have to have a long time of starvation. So if in one season the host will not come, this colony is going to die, because as far as we know, Simids are not able to survive like 2 seasons without getting blood meal. Okay, so I think there are following reason behind this pattern. There are more stable bat colonies in Northern Europe than in the south. With stable, I mean fluctuating, decide just to go to a different place for one summer. And the bats using mostly caves in Southern Europe, the caves they are using are too extreme with respect the temperature and humidity for the simids. And human dwellings provide a more moderate climate which is necessary for the simids. And maybe this also explains a little bit the puzzle how the human bedbugs came to the humans. Because in the Ice Age, the caves in southern Europe got too cold for the bugs. And the bugs were kind of leaving the caves with the bats, and at that time the humans started to have their first settlements, and that is maybe that we, the human, got our bedbugs in this time, not from caves, but from settlements. Okay, the next example I want to give from the mismatch between host and parasites is from Primicimix or Primicimix. That is really a rare bat. Here you see all records from this species, and you can see the green one are the most recent ones. And the reddish one are historical records, and for some places has been checked, and this species is not there anymore. Okay. A restricted distribution, but the host here is the Mexican free-tailed bat. You know that this one kind of makes colonies from several millions, and is a touristic attraction to see how the bats comes out in southern US. And in green, you can see the distribution of the host. It is distributed all over South America and Central America and the southern part of North America. But only in the red area you find the host, the parasite. And it's also important to know that some of the bats from South US, they stay over winter in South America, so they travel up and down. So it was actually easy for the bats just to go with them, but they do not. And I found interesting here, even if I do not know why, you have kind of two gaps of the distribution. One is what I will call is a biogeographical gap. It is an interesting phenomenon that simids, you find 14 species in Northern America, about 10 species in Southern America, but in the middle you have just 5, maybe 6 single records from 4 species. So in Central America, we just do not find simids, let alone those living on humans. And already Usinger was trying to fill this gap, but no, they are not there. Anyway, in this case, the Primicimix is not going over this gap with this host. But also on more regional scale, here you see we have a kind of a gap. Here you see a map from southern Texas, and you can see all the blue spots are caves where you have this Mexican free-tailed bat. And only in 3 of them we have found this Primicimix. So also even on regional scale, this parasite is not going with this host for whatever reasons. Okay, you can say, yeah, it's difficult to find these bedbugs in caves. Yes, it is. But I would say 10 of these caves has been intensively looked for this bug and haven't been found. Okay, so mismatch between hosts and parasites. I gave 2 examples. I could give more. The reason why is, of course, difficult to understand and to explain. Okay. Finally, I am lost. Okay. So I can continue. If it comes to population genetic studies, just lost, gone with this unknown host. And yes, so the main key to think about is how simids have to travel, right? Ah, yeah, now we are here again. Is the PDF now? Yeah, okay, doesn't matter. Yeah, down. Yeah, one more. Okay, here we are. Okay, can still use this. Yeah, okay. So I wanted to give you 3 examples for population genetic studies, but for introduce the problematic, I will remind you that, okay, so these secondary winglets, simicids, they have to hitchhike with their host. And for them, it's not so easy because they do not have such strong legs. So usually they sit on the wings of bats, or if you see in the one picture, on the foot of a bird. So, and yes, they hitchhike. We know that from the study from the cliff birds shown in this graph, if you remove all simicids by insecticides, within 1 month the population density is the same. So, and that study has been done like this, that only the adults has been counted, so there is a transmission back to the colony from other colonies. And usually it is this kind of, if you catch a bat, you usually find only 1 or 2 simicids on this back, not many. Sometimes the highest number was 9, but usually it's 1 or 2, and that is often females. So the whole system has to do with, you have single female colonization events, which then causing kind of forming a new colony. But there's also other pictures. The simicids you can catch on a bat, it's not kind of even distribution between species. You can easier find a Catcodemus than a Cimex pipistrelli, and hardly find any Cimex lectularius. Also, if it comes to the bat species, they are kind of differently preferred as a host for traveling. And it might be also a regional aspect about this. For example, in Bavaria, from 400 caught bats, more than 10 had simids on it, and in Bulgaria, from 2,000 bats, none of them had a simid on it. Okay, the studies we have one is from Cimex lectularius from cities, and what we know from there is kind of all individuals in one flat are kind of one family. They are related. Individuals from different floors and buildings are not related, let alone those from different part of the cities or different cities. So what we know, we have many independent colonizations, and our human bedbugs, they are even adapted against this inbreeding effect that you will have. The second study, what I know, is from Cimex adjunctus, which is a cimicid living on bats in North America, and what you can hardly see here is they have a stronger genetic structure than lectularius, but comparing to the host, they do not have such strong, the host has even a wider genetic structure. It's not shown here, but this is showing, okay, these are not so mobile as the host, the bats, which is not a surprise, but they are more mobile as the human bedbugs, or other way around. There's less random colonization aspects. Okay. When I was in the Sonora Desert with my colleagues to find this Hesperocimex for our phylogeny, we thought, okay, this kind of cacti where the Hesperocimex lives reminds us on building. Why not do a study like how is a natural different building, like different cactus population genetic here. Yeah, okay. And I have to, so the woodpecker, they're making the holes in the cacti, and this bug is the parasite of the woodpecker, and then they stay there and parasiting the woodpecker. But it's more complicated because of a lot of other birds and other animals using these holes for breeding or staying there. Okay, what we did, we opened the holes of some cacti only if we were sure that there's no bird breeding in that at the time, because we didn't want to destroy these nests. And then, yeah, we took out what you find on the bottom of these holes, and then we counted the bedbugs that we found. And the most of the holes do not have bedbugs. Those which have, they have small population between 5 and 50. We did an analysis of the blood meal, and we found out that 3 of different woodpeckers, cactus woodpeckers, were the host, even outside the breeding season. So it's not so bad for the simicids. Outside the breeding season, a lot of birds or woodpeckers come there to stay overnight. We had developed 10 microsatellites, and we made a kind of a genetic analysis from 5 to 12 individuals. So the first result I got yesterday in the evening, which should answer, okay, how are the simicids from different cacti are related? And that's kind of the first result of the haplotype network. From this, the empty things of cacti, we didn't find any bed bugs, and the colored one showing the haplotypes, and I'm not going to in detail that, but it looks like we have not so many haplotypes, only few, and suggesting also kind of single colonization events. So I think I'm running out of time, right? So I want just to thank my collaborators and the people founded us, and then I think I skip the take-home message because I do not have time for it.
Actually, we have one quick question. We have time for it.
Okay. Oh, yeah. Okay.
Thank you. For the bugs that colonize buildings, do you know if there is gene flow between various buildings and various families?
Very little. It is kind of to stay in the flat, and you do not walk around much. It is also would mean we do not know so much about it, but probably not. Stay just in one flat there. As long as there are humans, I think humans. If there are no people, they starting crawling around. Like we know also from simicids. We also know from simicids living on birds or beds, and especially in beds. If the beds are gone in summer and the bugs getting hungry, they're coming in the sleeping rooms of the people and looking for. But more or less they stay.
So the population then not suffer from bottlenecks?
Yeah, as I said, Simis lectularius is a paper out showing that inbreeding has almost no effect on them. Even outbreeding has a negative effect for their reproductive outcomes. So they are really adapted to the system. Okay.
Thank you. Now the last talk before the coffee break is a morphometrics of Fimata, splitting species by Pablo Varela.
Hi. Whoa. Sorry. Yeah. Good morning, everyone, and welcome to my presentations. Once again, my name is Pablo, and I would like to thank you all for coming here today. This presentation will focus on the morphometric analysis of a specific species, which is Fimata chilensis. I would like to apologize beforehand for my English. It's very difficult for me. Okay. First of all, I would like to introduce the characteristics and traits of the genus. Fimata is the most species genus with the Fimatini and has a complex taxonomy because of the species are polytypical, polymorphic, have sexual dimorphism, the specific delimitation is based on a few morphological characteristics, and for some species, only one sex is known. One important group defined by Kormilev is the Carinata group. Native from South America, is made up of 4 species. Theoretically, the 4 species share the traits and are defined by, in the head, the frontal process, in the pronotum, the lateral and angles and lateral notch, and head of the paramerum. Finally, they have a variable abdomen. One of these species is Fimata chilensis. Original name as Fimata carinata is widely found in southern South America and is the most abundant in Argentina. According to Kormilev, it is divided into 6 subspecies, each with its characteristic distribution. Fimata chilensis, from Peru to Chile. Fimata chilensis morrosi, from Bolivia and the northwest Argentina. Fimata chilensis cordobensis, from central Argentina. Fimata chilensis pampeana, from central and northern Patagonia. Fimata chilensis uruguayensis, from northeastern Argentina, Paraguay and Uruguay. And Fimata chilensis brasiliensis, from Brazil. This subspecies was discarded from the present study due to lack of access to material. So, the 5 subspecies in consideration are characterized by gradual difference in size. With the subspecies monrosi being the smallest, then chilensis and cordobensis with an intermediate and similar size between them, and finally, Pampeana and Uruguayensis are the larger ones. It is worthy to remark that Kormilev also mentioned the existence of dwarf variants for subspecies cordobensis and of giant variants for Uruguayensis. The criteria for separate the subspecies is the shape of the tip, elevation and length of the frontal processes, the shape of the lateral edge of the pronotum, mainly the angularity of the posterior lobe, and it is a more or less developed tooth, and the shape of the lateral angles of the abdomen, and the angle formed by segments 4 and 5. These criteria apply to males and are not entirely clear for females. Kormilev also mentions a gradual change in the length of the frontal process correlated to the angularity of the posterior lobe of the pronotum, and that of the lateral angles of the abdomen is related to the latitude. Among these variations, those of the pronotum are considered the most important and the assignment of the subspecific categories, although each description is accompanied by a characteristic of the connexivum, which is generally defined as variable. Taking into account the existence of subspecies, as well as the existence of intermediate forms and the dwarf and giant forms, in addition to the incorporation of new specimens outside the original distribution areas, the hypothesis for classification postulated by Kormilev become conflicted. The means that is not clear which is the criteria for the delimitation at the different taxonomic levels and what is the variation. That is to say, what do we use to separate the species? To test Kormilev's hypothesis of the subspecies of Fimata chilensis, we analyzed the variation in pronotum and connexivum shapes, removing the influence of size. For this, we use the geometric morphometric approach and the proxy screw validation based on the correct classification rates. We use this approach because it uses Procustes analysis to compare the shape of different specimens after a process of scaling, translation, and rotation that eliminates any possible difference or size effects. The cross-validation process will be used to assess the efficiency of the classification hypothesis. The study was carried out using specimens for the main Argentinian collections. Each specimen was sexes identified as a specific level following Kormilev criteria. 2 photosets were taken for each specimen, 1 for the pronotum and 1 for the connexivum, following the same alignment criteria for each specimen, using the same camera and the same scale to photograph each structure, creating 2 sets of photodata. Each set we worked with the TPS software series to digitalize landmarks and semi-landmarks on each photograph. Then the curves were aligned using the main chord bending energy criterion, and the centroid sides were also extracted. The data obtained were exported to Morpho-JSOFT, where the Procustes adjustment was performed and Procustes ANOVA was carried out to reduce alignment and digitalization errors of the landmark points. Based on this, the datasets were divided by sex and by structure. The analysis of the pronotum and the connexivum were carried out separately. From each Procustes setting and correlation matrix was obtained and on this normal principal component. Analysis and a pooled within group regression between the Procustes coordinates representing shape and center on side was carried out. We performed a canonical analysis of variance, and by this we obtained in the correct classification rates. To do so, we used the PAS4 software. We selected the structure with the highest percentage of correct classification rate, and based on this result, we changed the classification criteria to subspecies according to the type of connexivum and re-evaluate the correct classification rates. The result obtained showed that in both sexes, the connexivum has a higher correct classification rate than the pronotum, and for both structures, it is higher for females. In all cases, we test the hypothesis of independence between shape and size from a pooled within regression group due to the difference in between species, and in case this effect size on shape no exceed 10%. We select the PCA of the male connexivum to show you the variation between subspecies, but analysis for the pronotum and for the female accompany the results. We observed on the x-axis PC1 containing the highest variability, around 65%, and on the vertical axis PC2, one with 15% of variability. One dot represents each specimen and its color, as shown in the reference, the subspecies assigned, accompanied also by the wire diagrams corresponding to the average shape of each of one in color with respect to the global average shape in yellow. The black dot represents Monroe specimens, which is clearly separated from the rest of the group and whose average shape is the most different from the global average shape. In green, we can see the Cordobensis specimens, in red, the Chilensis specimens, in blue, the Pampeana specimens, and the dots turquoise, those specimens considered to be intermediate between the Pampeana and Cordobensis subspecies. The wire diagrams from this group shows a great similarity between them and little to no difference with respect to the global form. Finally, in orange, we can see the points corresponding to Uruguayensis specimens, and in purple, the ones between the Uruguayensis and Pampeana subspecies. The corresponding wire diagram shows a difference with respect to the average shapes, although more subtle than shown Win Monrosi and the opposite end. Despite this, we can see that the dots seem to be distributed in two groups. One made up the green, red, and most of the turquoise dots, the Chilensis and Cordobensis subspecies. The other group is made up of blue, orange, and purple dots, Pampeana and Uruguayensis subspecies. In all cases, Monrosi obtained an 100% of classification correct rates and significant Procus distance. For the other subspecies was variable according to sex to structure, mainly in intermediate subspecies. Uruguayensis was separated in females but not in males. The shape size correlation graph shows the existence of a particular shapes for the Monrosi specimens. For the rest of the subspecimens, they are a tendency to group Cordobensis and Chilensis, the green and red dots, and most of the intermediate specimens. They occupy the same shape space independently of center site. The trend is similar for the blue dots and for the violet and orange ones. This could also be interpreted as a shape gradient. However, when we implement the assignment to specific categories, uses only the connexiva, we observe that the rate of the classification increased considerably, reaching 95%, and that the tendency of separation between the mentioned groups increases. This result suggests that the existence of intermediate forms as a morphological gradient is an effect of using the pronotum of the mainly classificatory structure, which has a lower correct classification rates. Based on this result, the decision to be taken imply elevating the subspecies Monrosi to a species category, since all the specimens assessed have a similar and homogeneous morphology, both for the frontal process, which are always low, wide and wider rounded tip, in comparison with the rest of subspecies. A lateral silhouette of the pronotum with a very low lateral notch, the posterior lobe in obtuse without teeth, and it has a particular different connexiva from the other specimens. For the rest of the group, we propose to complement the analysis with molecular methods to evaluate the possibility of the existence of two groups within Fimata chilensis subspecies complex, or even to test if subspecies uruguayensis is also another species, since besides presenting a subtly different connexivum, the lateral silhouette also different slightly from the average shape, with a very marked angularity and a prominent tooth as seen in the silhouette below each diagram of the connexivum, for each subspecies in particular. Finally, we believe that the connexivum is the most important trait to separate these species, but it should be evaluated in conjunction with the pronotum. We also suggest extending this approach to other groups, as this problem is observed in other species with the Fimata, such as Fimata communis and Fimata fortificata, the latter of which has 7 subspecies. I would particularly like to thank Cecilia Morgan, Ariel Fernandez, and Valentina Castro Huertas for their help and encouragement, and to all of you for your attention. I'm not ready for questions.
Your English? Questions?
Hey, great talk and great project. Thank you very much. So I have a quick question on the Brazilian subspecies. Do you think that's a separate species altogether?
Sorry, creo que necesitar un intérprete.
Dice si brasiliensis es otra especie.
I think this brasiliensis is a more close to Fimata prestans, very similar, and in the definition of brasiliensis is about one male and on separately distribution, and no sense for me. But I think this is a thematic preference.
Any other question? Okay, thank you, Pablo.
Thank you.
So now your attention for two short announcements. Those living here in the Residencia de Investigadors who check out today, last time is noon, in an hour, please, because they have absolutely full up all the rooms, and so if you check out later, that's a mess for them. And the other announcement is that unfortunately it seems that the photographer is not satisfied with the result, and not because of you, of course. All of you are handsome and smiling, but there was something which he's not satisfied, and so he asks from you to bother again and try to do another picture today, same time, same place. So he gives thanks for that because he phoned yesterday very late at 11 or so. What happens? I'm not satisfied with the picture, but it looks okay on the mobile. No, but when you look in the screen, it's not as good as it could be. So give him a second chance, and thank you very much for that. Yes? No, you have to hurry up. Yeah, you have to keep the hour, yes. The picture will be, let's say, now we know where it is, so we leave the lunch venue, let's say at 2:30, and then it's a minute to take the place, to take the picture, and to arrive here. It means if we arrive 5 minutes later here, it's not that important, but we keep the schedule, yes. Okay, thank you. Ready for the coffee break?